Cell membranes are crucial to the life of the cell. Despite their differing functions, all biological membranes have a common general structure. Each is a very thin film of lipid and protein molecules, held together mainly by non-covalent interactions. Cell membranes are dynamic, fluid structures, and most of their molecules have about in the plane of the membrane. This lipid bilayer provides the basic fluid structure of the membrane and serves as a relatively impermeable barrier to the passages of most water-soluble molecules. The lipid bilayer provides the basic structure of all cell membranes. It is easily seen by electron microscopy, and its structure is attributable exclusively to the special properties of the lipid molecules, which assemble spontaneously into bilayers even under simple artificial conditions. Cell membranes can be classified in three categories. First, those that have more lipids than proteins. For example, cells that require isolation for electrical impulses, such as neurons. Second, those that have the same amount of lipids and proteins. For example, cells that their main function is transportation and metabolic. Third, those that have more proteins than lipids, for example, structures that have a great metabolic activity, such as the mitochondria. All of the lipid molecules in cell membranes are amphiphilic, that is, they have a hydrophilic or polar end and a hydrophobic or nonpolar end. The most abundant membrane lipid are the phospholipids. These have a polar head group and two hydrophobic hydrocarbon tails. In animal, plant, and bacterial cells, the tails are usually fatty acids, and they can differ in length. One tail typically has one or more cis-double bonds. As shown in this figure, each cis-double bond creates a small kink in the tail. Differences in the length and saturation of the fatty acid tails influence how phospholipid molecules pack against one another. The main phospholipids in most animal cell membranes are the phosphoglycerides. Another important phospholipid is called sphingomyelin. Together, the phospholipids phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, and sphingomyelin constitute more than half of the mass of the lipid in most mammalian cell membranes. In addition to phospholipids, the lipid bilayer in many cell membranes contain cholesterol and glycolipids. Eukaryotic plasma membranes contain especially large amount of cholesterol, up to 1 molecule for every phospholipid molecule. The lipid shape is a very convenient concept, frequently used to describe the volume occupied by phospholipids. Using this approach, phospholipids can be classed as cylinders, cones, and inverted cones, depending on the relative volumes of their polar head groups and fatty acid chains. The supramolecular organization or packing of such molecules originates the widespread bilayer structure and the non-lamellar H1 and H2 phases. The lipid composition of the two monolayer of the lipid bilayer in main membranes are strikingly different. Lipid asymmetry is functionally important, especially in converting extracellular signals into intracellular ones. Many cytosolic proteins bind to specific lipid head groups found in the cytosolic monolayer of the lipid bilayer. The enzyme protein kinase, for example, is activated in response to various extracellular signals. It binds to the cytosolic phase of the plasma membrane, where phosphatidylserine is concentrated, and requires this negatively charged phospholipid for its activity. Animals exploit the phospholipid asymmetry of their plasma membranes to distinguish between live and dead cells. When animal cells undergo apoptosis, phosphatidylserine, which is normally confined to the cytosolic monolayer of the plasma membrane lipid bilayer, rapidly translocates to the extracellular monolayer. The phosphatidylserine exposed on the cells of a cell's neighboring cells, such as macrophages, to phagocyte the dead cell and digest it. The translocation of the phosphatidylserine in apoptotic cell is thought to occur by two mechanisms. One, the phospholipid translocator that normally transports this lipid from the non-systolic monolayer to the systolic monolayer is inactivated. A scramblase that transfers phospholipids non-specifically in both directions between the two monolayers is activated. Sugar-containing lipid molecules, called glycolipids, found exclusively in the non-cytosolic monolayer of the lipid bilayer, have the most extreme asymmetry in their membrane distribution. Glycolipids probably occur in all animal cell plasma membranes, where they generally constitute about 5% of the lipid molecules in the outer monolayer. Hints as to the function of the glycolipids come from their location. In the plasma membrane of epithelial cells, for example, glycolipids are confined to the exposed apical surface, where they may help to protect the membrane against the harsh conditions frequently found there.